My name is Raina Maier, and I'm a professor of environmental microbiology in the Department of Soil, Water, and Environmental Science at the University of Arizona. One of the projects that I've worked on since I joined the University of Arizona over 20 years ago is examining a class of compounds made by bacteria called biosurfactants. And these are soap-like molecules. But what's really fascinating about them is their structural complexity and diversity. Bacteria appeared on Earth about 4 billion years ago. They've really driven the development of Earth and all of its ecosystems, going from a formerly reducing atmosphere to our oxygen-containing atmosphere we have now. And that was driven by developments made by bacteria. The reason I got interested in studying microbial surfactants, or soaps, or as I call them, biosurfactants, is from an environmental perspective. To see whether these materials could help us clean up sites that are polluted with organic and metal-containing wastes. In the mid-1990s, we discovered that these surfactants could complex metals very, very well. And so we have been since exploring these materials to actually remove metals from soil. And in our latest iteration, we're exploring urban mining. So if you have water coming from a waste treatment plant or water coming from a mining operation, can you use biosurfactants to recover very low levels of critical materials like yttrium or neodymium, and yet these metals are very, very important. Our cell phones have 60 different elements in them, and all of them have to be mined. And if we can use biosurfactants to recover low levels of these materials from waste streams, we'll be far ahead of the game in terms of supplying these critical materials. Another discovery that I've made during my career with Mike Stangolini, who was then in plant pathology, was that biosurfactants have potent activity against a class of fungi called zoosporic plant pathogens. This is the pathogen that caused the famous Irish potato famine. And we actually patented the use of a biosurfactant called rhamnolipid for use against these zoosporic plant pathogens. One of the big advantages of working at the University of Arizona is that there are few to no barriers to working between departments and between colleges. I have been able to work with the College of Pharmacy, the Department of Chemistry, Department of Plant Pathology, and I've only gotten encouragement to form these collaborations. And I think it speaks volumes for the University of Arizona that we're allowed this kind of freedom, because you don't find that in a lot of universities. This is the first set of biodegradation experiments that we're running on the synthetic biosurfactants that we're exploring making in our Tech Launch Arizona grant. So as you can see, we've got these very interesting flasks where we put our biosurfactant on the right-hand side along with a sewage sludge inoculum to provide biodegrading bacteria. And on the left-hand side, we have a solution of a basic solution that collects any CO2 that shows metabolism of the biosurfactant. So when the biosurfactant is biodegraded, it produces CO2, which we collect, and so we can quantify the biodegradation of these synthetic analogs of our biosurfactants. And we're hoping they're biodegradable so that we can use them as analogs for the biosurfactants themselves. So these biosurfactants are a platform on which we can make discoveries for new applications and also discover whole new biosurfactants that can be explored for new applications.